Okay, let me invite you to this discussion on immunology. In this set of discussion, we'll be looking at T cells and cell-mediated immunity. So we'll begin by trying to introduce ourselves to what T cells are. So T cells, otherwise called T lymphocytes, they are a particular type of white blood cells which play a central role in cell-mediated immunity. So they are a type of white blood cell, and they have a critical role to play in this type or branch of the immune system that we call cell-mediated immunity. So by doing that, by playing that central role in cell-mediated immunity, they help eventually to defend the body against intracellular pathogens. So the pathogens that are found inside the cell. Okay. So those are T cells. But now we've seen that these T cells play a critical role in cell-mediated immunity. So the next question then becomes, what is this cell-mediated immunity? So cell-mediated immunity is a branch of the immune system which happens not to rely on antibodies, but instead involves T cells to directly attack either cells that are abnormal or infected. So with the cell-mediated immunity, you have T cells which are mobilized to attack cells that are infected or cells that are abnormal. So with infection, then we talk about pathogens. But with abnormal cells, then there is a possibility of cancer, the cells being cancerous. Okay? So this cell-mediated immunity is crucial for eliminating intracellular pathogens, the pathogens that are found inside cells, because remember, it is attacking a cell that is infected. So when a cell is infected, the pathogen will be in the cell. So if cell-mediated immunity is mobilizing T cells to attack infected cells, then by doing so, by attacking these infected cells, it can help to eliminate whatever it is inside these cells. Okay, so by eliminating the intracellular pathogens, then cell-mediated immunity will help to prevent the spread of infections. Because if T cells are mobilized to attack infected cells, then if the attack is successful, then the cells that are infected will be destroyed. And by doing so, then the infection in the cells cannot spread. So cell-mediated immunity can then help to prevent a spread of these infections. So cell-mediated immunity, which in this case we also abbreviating as CMI, is primarily responsible for targeting cells that are infected with viruses, cells that have tumor, so the tumor cells, and the intracellular bacteria. So you are talking of a cell that has a pathogen inside it, or a cell that is abnormal. But now, where do these T cells come from? So T cells originate in the bone marrow. So in the bone marrow, they originate as immature cells. But then they move to the thymus gland, where they now mature. And as they mature, they now learn to distinguish between what is self and what is not self. So what is self is what belongs to that body. So your T cells have been trained to identify what belongs to you and what belongs to another person. So what is foreign? Anything that is not part of you will be foreign, be it a pathogen or any organ, if someone was to even donate blood or anything. So the T cells, they originate in the bone marrow, but then they move to the thymus gland where they mature. And as they mature, they are now trained on how to distinguish between self antigens and non-self antigens. We'll look at that more, most likely the next video. So the main functions of T cells, there are three. One is to recognize the infected or abnormal cells. So the first thing is they recognize either cells that are infected or cells that are abnormal. Then the second function is to destroy the infected cells. So as the T cells destroy these infected cells, they end up now preventing the spread of infections because they've destroyed the cells that had that infection. Then the third function is they regulate the immune response so that there is no overreaction. Okay, so they help in regulating the immune response. So what are the types of T cells and what are their respective functions? So we've got around 4 types of T cells. The first type is the helper T cell or the CD4 T cells. These ones, they activate other immune cells. So they activate other immune cells. Other immune cells in this case include B cells, the cytotoxic T cells, and these immune cells are activated when the helper T cells release cytokines. Okay. So helper T cells activate immune cells by releasing cytokines. The next type of T cells is the cytotoxic T cells, otherwise called the CD8 T cell. So what the cytotoxic T cells do is they kill cells that are either infected or cancerous. Okay. So they kill either infected or cancerous cells, and they do so through the release of toxic molecules. Okay. Then the third type are the regulatory T cells. Remember, one function of the T cells was to regulate the immune system. So the regulatory T cells actually now do this function. They suppress the immune response. They suppress the immune response. So when they suppress that, then they prevent an excessive reaction. Then the last type of the T cells are the memory T cells. One function of the immune system is supposed to remember what you've been infected with has to be remembered, so that when there is a reinfection, when it infects you again, there should be a faster response. So the type of T cells that helps in remembering the past infections, so that there can be a faster response when there is a reinfection, are the memory T cells. So how exactly, through which steps, does the cell-mediated immunity work? So the first step, step 1, is antigen presentation. So when a foreign pathogen has infected a cell, okay, when a foreign pathogen has infected a cell, there is what we call the antigen presenting cells. They process that pathogen and they now present fragments of that pathogen, which is now antigen, they present them on the surface. Okay, they present them on the surface of that particular cell. So a cell has been infected by a pathogen. Then that pathogen is picked up by the antigen presenting cells. So that pathogen, after being picked up by these antigen presenting cells, they process that pathogen. Then what they do after that, they now present fragments of this pathogen, which we are now calling the antigen. So they present the fragments of this pathogen on the surface of that cell. And what they use to present these fragments is what we call the major histocompatibility complex, or just in this case the MHC complex. We'll look at the MHC complex next. So MHC complex happens in two categories, two types, MHC class 1 and MHC class 2. So MHC class 1 interacts with the cytotoxic T cells. Remember, that is now dealing with infected body cells. Then MHC class 2 interacts with the CD4 helper T cells. That is for immune coordination. So antigen presentation. You have a pathogen which has infected a cell. Okay. Then once that pathogen has infected a cell, is now picked by the antigen presenting cells. The antigen presenting cells are things like macrophages or dendritic cells. These have the ability to swallow. Okay, they have the ability to swallow this pathogen. Okay, so the phagocytosis. These are cells that have the ability to phagocytose a pathogen. So once they've done that, then they'll present fragments of that pathogen on the cell surface, on the surface of that cell which had been infected by that pathogen. What they use to present the fragments of that antigen or fragments of that pathogen are now the MHC molecules, or the major histocompatibility complex molecules. Then now the second step is the activation of the T cells. Okay, the activation of the T cells. So the CD4 helper T cells will recognize the MHC class 2 and a complex between the MHC class 2 and an antigen. So the MHC class 2 will be having an antigen attached on it. Okay, so the CD4 helper T cells will recognize that. MHC is what is presenting the antigens on the cell that is infected by that pathogen. Okay, so the CD4 helper T cells will recognize the MHC class 2 and the antigen that it will be presenting, so that complex. So once the CD4 helper T cells recognize that, then they will release cytokines. So once the CD4 helper cells have recognized the MHC class 2 and the antigen, the complex that the MHC class 2 and the antigen have formed, once the CD4 helper T cells recognize that complex, then they release cytokines. So those cytokines are the ones that will activate other immune cells. Then the CD8, the cytotoxic T cells, these ones recognize a complex between MHC class 1, the major histocompatibility complex 1, and the antigen. So CD4 is MHC class 2 and the antigen. CD8 is MHC class 1 and the antigen. So once the CD8 cytotoxic T cells have recognized this complex between MHC1 and the antigen, they now become activated and they begin to attack the infected cells. Okay? So let's have a go again at that activation bit. The CD4 helper T cells, they are recognizing a complex between MHC class 2 and the antigen. Once they have recognized that complex, then they will release cytokines to go and activate other immune cells. But the CD8 cytotoxic T cells will recognize a complex between MHC class 1 and the antigen. So once they've recognized that complex, the CD8 themselves as T cells, they will become activated and they begin to attack infected cells. The CD4 activates other immune cells. The CD8 will begin to attack infected cells. So let's now see how then the T cell responds. That's now the step 3. So the response of the T cells. One, the cytotoxic T cells will now kill infected cells. Okay, it will kill the infected cells. How does it do the killing? One, it punches holes on the cell membrane of the infected cells. So it releases perforins. The perforins will now begin to make holes on the membrane of the infected cell. Then the other mechanism through which it can kill these infected cells is by cell death, the programmed cell death, what we call the apoptosis. Okay, so it will release the granzymes. Okay, it will release granzymes which will now enter. These granzymes will enter the infected cell. Once the granzymes have entered the infected cell, they will trigger a programmed cell death. Okay. The second way through which the T cells respond is that the helper T cells will now boost the immune system. Remember, when the CD4, they were activating other immune cells. So B cells will be activated. So they are activated to produce antibodies. Okay, they will be stimulating macrophages. Macrophages are the ones that can engulf pathogens. Okay, then more immune cells will be recruited so that they can help in fighting the infection. So within the body, an infection ni kama majambazi. Okay, so it has come to attack the body. So your body has to recruit a lot of soldiers to help it fight. Then now the step 4 is the immune memory. This is where now we have the memory T cells. So some of those T cells will then now convert and become memory T cells after the infection. So these memory T cells are the ones which will now be used in case the same pathogen invades or attacks you again. So if the same pathogen invades again, the memory T cells will recognize it. Okay, so once they recognize it, then they mobilize a stronger prevention immune mechanism so that it can prevent reinfection. Okay, so those are the 4 steps through which this cell-mediated immunity works. So the first is antigen presentation. The second is T cell activation. The third is T cell response. And the fourth is the formation of the memory T cells. Okay. So what is the importance of these T cells, both in the maintenance of health and in the occurrence of disease? So the first importance is they help in protection against infection, because they help in fight. They are essential for fighting against viruses. Remember, they are attacking cells that have been infected by viruses. Okay. The other bit is they also help in cancer immunity because they help in destroying abnormal cells. They help in destroying abnormal cells, so they can create cancer immunity. But at times, when the T cells, when the cell-mediated immunity overreacts, when the T cells, the reaction of the T cells, when it happens beyond the limits, then it can attack the normal body cells. So when your own immune system is attacking your own body, then it creates what we call the autoimmune diseases. Okay. The fourth importance is the interaction between HIV, a virus, and these T cells. So the CD4 T cells are the ones which are infected by HIV. So HIV targets the CD4 T cells. And once it targets them, it now ends up weakening the body immune system. And if not treated using ARVs, they now end up leading to the development of AIDS. The last importance of the T cells is transplant rejection. So remember, there was an aspect of these T cells recognizing or differentiating between what is foreign and what is not foreign. So by doing that, they will recognize transplanted organs as foreign, and therefore they will attack them. So if something is foreign, they attack it. By attacking it, they now lead to a rejection. Okay. So in summary, we've seen that T cells are a type of white blood cell, and they are crucial for the cell-mediated immunity. Helper T cells, otherwise called CD4, they help to activate other immune cells. How they do the activation is by the release of cytokines. Then the cytotoxic T cells, or the CD8, these ones help in killing cells that are either abnormal or infected with pathogens. Then our cell-mediated immunity is an immune response that relies on T cells. It does not rely on antibodies, but it relies on T cells. So cell-mediated immunity relies on T cells to eliminate intracellular threats. Then now the development of the immune memory is when memory T cells will remember whatever it is that has infected you in the past, so that whenever there is a reinfection, there can be a faster response. Okay? That's about T cells and cell-mediated immunity. So next, let's look at now the receptors of these T cells. So the T cell receptors and what we've been mentioning as the major histocompatibility complex. So the immune system serves as the defense mechanism of the body. It defends the body against harmful pathogens. We've seen that a critical component of this defense, of this immune system, is the cell-mediated immunity. This cell-mediated immunity is critical, and it relies largely on T cells. So these T cells will recognize and eliminate threats. Okay. It will recognize and eliminate threats. But now, before the T cells work, they have to be activated. Before the T cells can recognize and eliminate pathogens or disease-causing microorganisms or cells that are abnormal, they must be activation. They must be activated, so they have to undergo activation. So the activation of these T cells is dependent on two essential molecular components. So two things that are critical, two things that the process of T cell activation depends on. One are the T cell receptors. Then two is now the major histocompatibility complex. The T cell receptors, these are specialized proteins which are found on T cells. Okay, so these specialized proteins that are found on T cells are the ones that will be recognizing the peptides, the proteins that are from antigens. Okay, so it means for the T cell to recognize an antigen, it uses these specialized proteins, which are now the T cell receptors. Then the major histocompatibility complex, otherwise abbreviated as MHC, this refers to a group of molecules. And what this group does, it helps in the display of the processed antigens on the surface of cells. Okay, so when the antigens are processed, they are now displayed on the surface of the cell by the MHC. It is once that they are displayed that now immune recognition and reaction can happen. Okay, so you can think of the MHC ni kama hiyo ndio sasa inabeba bendera juu, because when you carry the flag up, that is when you are able to recognize that there is something here. So the MHC is what now displays the antigens, and these antigens are displayed on the surface of cells. Okay, so it is therefore the interaction between the T cell receptors and the major histocompatibility complex, the MHC molecules. That interaction is fundamental for the immune response. Because once the T cell receptors, which are found on T cells, once they interact with the MHC molecules, then the T cells can now be able to detect whatever is infected, whatever is abnormal. Once the T cells have detected either something that is abnormal or infected, then they can now initiate the appropriate immune reaction. So let's look at now the T cell receptors. So as we've said, these are proteins that are expressed on the surface of the T cells, and they play a crucial role in immune surveillance, because the T cells now use these proteins to check around to identify the antigens, okay, to identify the peptides that are specific to particular antigens. Now, these peptides will have been presented by major histocompatibility complex molecules. And these peptides will have been presented by the MHC molecules on the surface of the cells. Okay. So T cell receptors are found on the surface of T cells. Their function is they help surveillance, ni kama kuangalia, kuzunguka, to check. Okay, so the T cell receptors help in checking, in trying to moving around to identify how specific any antigenic peptides that are presented by MHC molecules. The MHC molecules will be presenting, the antigens are created now by pathogens or any abnormal cell. So the antigens will be what was found inside the cell. Hiyo ndio inachukuliwa, then inakuwa presented on the surface of that cell. So if there was, for example, an infection inside, if there was an infection, a pathogen inside that cell, then the proteins of that pathogen are the ones that will be picked by the MHC and presented on the surface of that cell. Then now the T cells will recognize those peptides on the surface of the cell which have been presented by the MHC, will recognize it as foreign. Then it initiates an immune reaction. But if the protein, the peptides that are being presented by the MHC are of the same cell, they are not foreign, then the T cells will recognize it as self. Then now it does not initiate an immune reaction. So by so doing, it then only reacts when something is foreign, but it does not react if something is not foreign. So what is the structure of this T cell receptor? So the T cell receptors are composed of two major protein chains. So you have the alpha chain and the beta chain. Then those chains are linked, are joined by a disulfide bond, and they contain these four. So they have a variable region, a constant region, they have a transmembrane region, and they have a cytoplasmic tail. Okay, a cytoplasmic tail. For the T cells to function properly, they also express the co-receptors. So these co-receptors will enhance the recognition of antigens. Okay, so they enhance the recognition of antigens. So next, let's now look at the major histocompatibility complex, the MHC. What is the MHC? So MHC is made up of a set of genes. These genes code for some surface proteins. Okay, and these surface proteins that are coded for by this set of genes are the ones that are responsible for presenting antigens. So an antigen is a marker, is unique. An antigen is like a flag. But what raises that flag up is now the MHC. That is what raises the flag. Okay? So the importance of these MHC molecules is it plays a very key role in the recognition of what is self and what is not self. So the antigens that are self and the antigens that are non-self. Okay. So if the antigens are from a cell of the body, then the immune system will recognize it as self. But if within that cell there was a pathogen, then the antigens will be from the pathogen. Then the immune system will recognize those antigens as non-self. The types of the MHC, they come in two types, MHC class 1 and MHC class 2. So MHC class 1 is expressed on all nucleated cells. Okay, is expressed on all nucleated cells. And what happens with it is that the MHC class 1 presents antigens to the cytotoxic cells or the CD8 cells. And the MHC class 1, it deals mainly with intracellular pathogens. So what is found inside, intracellular, inside a cell. So it presents antigens to CD8, that is MHC class 1, CD8 cytotoxic cells. The type of antigens that are presented to the CD8 are intracellular pathogens. So you are talking of viruses or other tumor proteins. Okay. The other type of MHC is MHC class 2. MHC class 2. So MHC class 2 is expressed on antigen presenting cells. So the antigen presenting cells include things like macrophages, dendritic cells, and B cells. Okay. So it means the MHC class 2 will be expressed, will be found on macrophages, on dendritic cells, and on B cells. Then the MHC class 2, it will present antigens to CD4 helper T cells. So MHC class 1 presents antigens to CD8 cytotoxic T cells, while MHC class 2 presents antigens to CD4 helper T cells. The antigens that are presented by MHC class 1 are intracellular pathogens. The antigens that are presented by MHC class 2 are extracellular pathogens. Okay, that's an important distinction to make. There is a bit of restriction with respect to the MHC, so that T cells can only recognize antigens when they are presented by the appropriate MHC. So for example, CD8 will only recognize antigens that are presented by the MHC class 1. That's the restriction. So if MHC class 2 presents antigens, CD8 cytotoxic T cells cannot recognize them. When the CD4 helper T cells will only recognize antigens that are presented by MHC class 2. So when the MHC class 2 presents antigens, then only the CD8 helper T cells will recognize them. Okay. When the MHC class 1 presents antigens, only the CD8 cytotoxic T cells will recognize them. So that specificity, that bit of restriction is now what ensures that cytotoxic T cells only focuses on intracellular infections, and the helper T cells will coordinate the response against extracellular pathogens. So that is the distinction. That is why only these deal with a certain type of infections, and these other type of T cells also deal with a different type of infection. So let's have a look at how the T cell receptors and the MHC interact, and how that leads to the activation of T cells. So the first step in this process is the processing of antigens. So in the MHC class 1, MHC1 pathway, that one deals with cells that are infected. So infected cells will process pathogens that are inside them, so intracellular pathogens. So doing that, they will degrade these pathogens into peptides. They now present these peptides on MHC1 molecules. Okay, so cells that are infected will process whatever infection, whatever pathogen that is inside those cells. Then in the process, they degrade those pathogens to give peptides. And those peptides will now be presented on the MHC1 molecules. The MHC2 pathway, this is now dealing with the APC, the antigen presenting cells. So with the MHC2 pathway, in it, the antigen presenting cells will engulf. Remember the MHC type 2, the class 2, it deals with the extracellular pathogens. So they are expressed on antigen-presenting cells. So the antigen-presenting cells will engulf the extracellular pathogens. To engulf is to sort of to swallow. So after swallowing these pathogens, they process them, then present the antigenic fragments on major histocompatibility class 2 molecules. So. Now that is the processing. But now the second step is the antigen presentation. So the MHC class 1 antigen complex is recognized by CD8 cytotoxic T cells. Then the MHC class 2 antigen complex is recognized by CD4 helper T cells. That's now the presentation. Remember when we were talking of the restriction, the specificity, this is what we were implying. Now the third stage is the T cell activation. So once a T cell binds to its specific MHC antigen complex, there will be some additional signals for full activation. Okay, so for the MHC class 1 with its antigen from the intracellular infections, once they bind to its T cell, then an additional signal is required before that T cell is fully activated. Then when the MHC class 2 antigen complex, so the MHC class 2 is presenting antigens from extracellular infections. So that complex between MHC and the antigens from the extracellular infections, that complex is what will be recognized by the CD4 helper T cells. So the CD4 helper T cells will bind to the MHC class 2 and its specific antigen complex. Okay. Now, once the signal that is required for full activation happens, then the activated cytotoxic T cell will now destroy the infected or abnormal cell. Then equally, the activated helper T cell will release cytokines so that they can enhance immune responses. So the function, either to destroy or to enhance immune responses, is happening after activation. But activation is happening after a T cell binds to its specific MHC class and antigen complex. That's when the activation happens. So it is important that a particular MHC type binds to its correct antigen. When that happens, then the required signal for full activation is initiated. Then once that happens, then now the T cells become fully activated. Then the cytotoxic one will now destroy the infected cells. Then the helper T cell that is now activated will release cytokines. Once these cytokines are released, they help in enhancing immune responses and also in the stimulation of other immune cells. So what is the importance of the T cell receptor and the major histocompatibility complex interaction, the interaction of these two with reference to health and disease? So one, it helps in the prevention against nest infections. Okay. So for example, when you are talking of viral infections, the MHC class 1 will prevent viral peptides, will present viral peptides to cytotoxic T cells. Then after they are activated, they will now eliminate the infected cells. So that will be preventing offering protection against viral infections. If you are talking of extracellular pathogens, then the MHC class 2 comes in. So like bacterial infections, you have the MHC class 2, which will present the fragments of these bacteria to helper T cells. Then these helper T cells will activate macrophages and B cells so that they can destroy these bacteria. The other importance is in cancer immunity. Okay, so cancer cells may present mutated proteins on MHC class 1. If a protein is mutated, then it will be recognized as foreign, so it will be eliminated. Okay, if a protein is mutated, the immune system will recognize it as a foreign antigen. Then that protein will now be eliminated. But there are some cases where you have the autoimmune diseases. As we indicated earlier, these ones happen when the T cells have overreacted. By doing that, they may attack self tissues mistakenly. So when they attack what is self as if it is non-self or it is foreign, then now it leads to a disease that your body is attacking itself. Okay, so it is called autoimmune diseases. The other importance is when we talk of the organ transplant. So the MHC are very polymorphic. So each person has unique MHC proteins. So if you get a transplant, an organ transplant, you are likely to have a mismatch from your donor. And if that happens, then your immune system will recognize that as foreign, and then it will reject it. So to prevent that rejection, people opt to suppress the immune system by using immunosuppressive drugs. Okay. Then the last application, the last importance, is their role in vaccines. So with vaccines, you are introducing a harmless antigen fragment. So you introduce a disease-causing microorganism that is not harmful, cannot cause disease. So once that disease-causing microorganism, which can't cause disease at that particular time, is introduced into your body, your cells will process it and present it using the MHC class 2 molecules to helper T cells. So once it is presented, the memory T cells will keep it in mind so that anytime there is such an infection, now a real infection, then a faster immune response can be initiated. Okay. A faster immune response can be initiated. So in summary, T cell receptors, they recognize antigenic peptides that are presented by MHC molecules. Then MHC molecules, they happen in two types, MHC class 1 and MHC class 2. So the MHC class 1 is found on all nucleated cells. And what the MHC class 1 does is that it presents antigens to CD8 cytotoxic T cells. MHC class 2 is found on antigen presenting cells. And what the MHC class 2 does is that it presents antigens to CD4 helper T cells. So the easiest way to remember, when you're talking of MHC class 1, so you have a CD8 and a CD4. So class 1 goes with a CD that has a higher number. So MHC class 1 has a CD8. It presents antigens to CD8 cytotoxic T cells. MHC class 2 presents antigens to a CD that has a lower number, so a CD4 helper T cells. Now the interaction between the T cell receptors and the MHC that interaction between the T cell receptor and the MHC is essential for activating T cells, because the correct MHC has to bind to a correct antigen before that T cell can now be activated. Then after the T cell has been activated, that's when now it can actually work against the what? The disease-causing microorganism, the pathogen or the antigen in that case. Okay? So that will mark the end of this. Read the notes, listen to the video, and should you have a question, you know what to do. You reach out and I'll be happy to engage again. Thank you very much, and looking forward to our next interaction. Asante.